Welcome to Shimadzu 7th Global Pharma Summit. The pharma industry today has grown through leaps and bounds and is the third biggest industry today with more than $1 trillion US. And today, the third fastest growing industry that is being used extensively to handle all our modern day challenges is artificial intelligence. In keeping up with these future trends, Shimadzu is the first and the only instrument company that has developed the groundbreaking technology of analytical intelligence. This combines artificial intelligence, Internet of Things, machine-to-machine protocols to make the instrumentation for pharma the most intuitive and intelligent ever. In fact, from now onwards, all Shimadzu instruments will be powered by analytical intelligence. It gives me immense pleasure to reveal to you that this year's Shimadzu Global Pharma Summit is themed Transforming the Future of Pharma Together, and it's being powered by AI or analytical intelligence. Shimadzu started to organize the Pharma Summit in 2007, and today we are in its 7th edition. We are the only instrument company to organize this kind of a global pharma event in a grand scale. And I'm very happy that this year we have reached the pinnacle of over 1,000 plus key industry leaders. This has been only possible because of all of you. Please give a big round of applause to each and every one of you. We have witnessed an increase in the demand for Shimadzu informatics over the past years. As you know, informatics is the key to ensuring that pharma is easily able to face regulatory challenges with confidence. This rising demand for informatics is the main reason why we have a dedicated session on data science today in the summit. For success in healthcare, ladies and gentlemen, we not only need great analytical and medical technologies, but we also need synergy between the analytical and medical technologies. And Shimadzu is the only company in the world that offers you this synergy to provide optimized healthcare for better healthcare as well. In fact, we are collaborating with an increasing number of innovators and pioneering partnerships for advanced healthcare using mass spectrometry and medical imaging to transform the future of pharma and healthcare. Innovation is in Shimadzu's DNA. Excellence in science is in our blood. And you, esteemed customers, you are our family. I'm happy to say that today, because of your valuable support, we are in the top 2 instrumentation company in the world. With that, ladies and gentlemen, I would like to conclude by saying, let us together transform the future of pharma together. Sit back and enjoy the summit. Thank you.
That was Mr. Prem Anand, Executive Officer and Senior General Manager, Shimadzu Asia Pacific. A big round of applause. Welcome, ladies and gentlemen. My name is Sandy. I am Deputy General Manager in Shimadzu Asia Pacific, Singapore, and I am your host today. The Shimadzu Global Pharma Summit offers the biggest platform for pharma opinion leaders to network, share, and discuss the strategies to transform the future of pharmaceutical industry. And today, we have more than 200 pharma leaders from more than 60 organizations and 20 countries present here. Welcome, everyone. Shimadzu, we understand the need of every market, and this Pharma Summit is our commitment to you, okay? So we hope that you'll utilize this platform to network, discuss, and share your strategies to transform the pharma, future of the pharma. Okay? Without further ado, let's begin the summit. And every year, we have some unique ways to open the summit. This year, we have a futuristic ball, which will transform the future. Okay? And to do the honors, may I invite some of the dignitaries, Mr. Vijay Kumar Singh, JB Chemicals, India, to please come on the stage. Dr. Uthai Suvankut, Thammasat University, Thailand, to please join the stage. May I also invite President and CEO of Shimadzu Corporation, Japan, Dr. Teruhisa Ueda, and Managing Director of Shimadzu Asia Pacific, Mr. Tetsuya Tanigaki, to join the stage as well. Welcome everyone on stage. So what's going to happen now? On count of 3, all 4 of you, please place your right hand palm on the futuristic ball. Okay? On the count of 3, ladies and gentlemen, please join me in the countdown. 3, 2, 1. Pharma Summit 2019. Ladies and gentlemen, throughout the day, you will not only learn the different aspects of pharma domain like data science, technology, and therapeutics, but you will also experience some unique pharma workflows that will transform the way you are working in your lab. So further, now I would request the Shimadzu legend, Dr. Teruhisa Ueda, who with his 37 years of dedicated service has transformed Shimadzu to a truly global organization. Dr. Ueda, please give us a strong opening speech. Thank you.
Thank you, Sandy, for your kind introduction. Good morning, ladies and gentlemen. I'm Teruhisa Ueda. President and CEO of Shimadzu Corporation. It's my great honor to welcome all of you to the 7th Shimadzu Global Pharma Summit 2019. Each and every member of Shimadzu family would like to thank you for your gracious presence here today at our flagship Pharma event. I'm very pleased to see so many people from so many countries on this very special occasion. The Pharma Summit brings together the most renowned key industry leaders from across the world to network, share, and discuss ways in which we all can together contribute to society through science and technologies. We take pride in the importance of working with our customers, researchers, and partners to support human health and the pharma industry. At the same time, we also recognize that society is currently undergoing major changes, and we are thus continuously improving ourselves to become an even stronger partner of innovation and technologies. The summit is a relation of this pioneering spirit. This year, the summit is in its 7th edition and is themed Transforming the Future of Pharma, as Prem mentioned. Since its inception in 2007, the summit has enabled the global pharma industries to better understand the latest trends and face challenges together with Shimadzu. As you are aware, most things in the world today are being powered by AI. The summit is also harnessing the power of what Shimadzu calls analytical intelligence. This only its kind innovation in the world will be the backbone of all the future labs in the world, and you will experience these disruptive technologies firsthand in this summit. The global pharma industries is among the top 5 industries in the world and is also the most profitable. All major pharmaceutical companies around the world are constantly evaluating their strategies and operational approaches to leverage on the current trends in pharma. Keeping this in mind, the Pharma Summit has always showcased the most important development in the industry. As you can see, the topics featured here today range from paperless lab to automation, and from biopharma to medical cannabis. You may know Shimadzu as just an analytical instrument company, but we also provide synergetic technologies and products with medical innovation. With the award-winning technologies that we provide, and from expertise and experience you possess, we'd like to together tackle the challenges that the pharma industry faces and make this world a better place. We understand that collaboration is the best way in which we can contribute, and so we are developing award-winning innovation with university and industry leaders like you from around the world. Excellent science is not only a motto, but a way of life in the Shimadzu family. And it is in this same spirit that we bring you the 7th Shimadzu Global Pharma Summit 2019. We hope that the network you built here today will contribute the advancement in your organization. We wish you a successful and fruitful time at the summit and a very pleasant stay in Singapore. Thank you very much.
Thank you very much, Ueda-san, for your enlightening talk. As you rightly highlighted, the audience will definitely experience the AI-powered technologies today. Ladies and gentlemen, this year, we also have a live streaming on YouTube of this mega event. As you can see the QR code here, you can scan the code and you can share with your colleagues, your customer or your counterparts so that they can also join the event virtually. Okay, this is another technology development that everybody is not possible to come here and join the event, but they can enjoy what is going on here. Okay, so everybody, please feel free to scan the barcode here. All right, so with that, let's start with the first session, that is the technology of data science. About 15 years back, data science, big data, and data analytics were not so popular terms in pharma. But recently, in last 5 to 7 years especially, it is becoming the integral part of pharma infrastructure. Data is indeed very important if it is managed well and interpreted correctly. During a drug development, terabytes of data is generated, and data science can greatly help to use this data to meaningful findings in much shorter times. In this session, we will hear different aspects of data science in pharma. Compliance and efficiency are always on compromise in pharma. And that's the reason the pharma companies are moving towards paperless lab to avoid manual errors and non-compliance. Our first speaker, Mr. Indrajit Bose, will share some of the insights on paperless lab. Mr. Bose is Vice President Corporate QA in Lupin Pharma, India, and he has been instrumental in implementing several OpEx initiatives in lab as well as manufacturing processes. Please welcome our first speaker, Mr. Indrajit Bose, for his talk on paperless lab, dream or reality. Mr. Bose, please.
Thank you, Sandy. Hello, am I audible? Yeah. Thank you, Sandy, and good morning, ladies and gentlemen, all the dignitaries and delegates. I'm privileged to be here, and really thanks Shimadzu team for organizing such a great summit here. Well, my topic is, paperless lab, which we always look for, whether it's a dream or reality. And let me tell you, we are in an area or in the era where we feel that this is quite a feasible thing. 20 minutes I have, very short time to cover such a big topic. So what I'll try to do is that I'll try to see that you get a vision that what kind of things we are expecting in the industry now, what is the regulators' expectations, and at the same time that to what extent it is possible, and some of the areas we feel the challenges are more there, and that's something I'd like to cover. So, yeah. So I just, whenever you are going to take up the journey to the paperless lab, you have to always keep in mind one thing, that what is the current regulatory lead? And there's the first topic is that the 20-hour CFR compliance. Now, even though it is not my domain right now to speak on that, but always keep it in mind that we are sometimes very much enthusiastic to implement, but then once during the audit or during our day-to-day work, we feel that there are challenges with respect to computer system validation. So that's a big topic, very complicated process, but at the same time, you have to always be careful that whenever you are implementing that, we need to assure ourselves that it is absolutely in a compliant zone. Then electronic submissions, and then the business needs. All us, we know that there is an enhanced compliance and quality. We have cost reduction, timely deliverance, efficient management, manufacturing lab operation, quick decision making. So this is just an idea about, on what is the different citations, repeat citations FDA has in the last 3 years. And if you really see, you will find that the discrepancies and the lab controls is really dominating. Now, many of the areas where the lab compliance, I must say that, we are still in a manual activity more than the other industries. So there are a lot of scope to automate that process, and that way we reduce many of the manual errors, and then really helps us in reducing the errors and the non-compliances. Now, this is, overall, if you look into, I've just divided into several zones, which actually the laboratory function is. It starts from sample management, right? Even with the raw material sample, when you receive in the laboratory, it has to come to the warehouse, and then there is a good receipt note will be made in the warehouse, and then the material comes to QC. Now, sometimes I see that the lab operation in the company is automated, but then the samples and all these activities get manual. At the end, you'll have to then put all this control on the sampling, the sample entry in the system, that becomes manual. So when you are going for a paperless journey, you have to make sure that you have a proper roadmap. And in many cases, the sample management can be absolutely automated. So what happens in the process is that you have a sample, you have a vendor, which is already there in your system. So it's an approved vendor. The material cannot come without the approved vendor in the system. And then you develop a system, which is quite possible now, that the moment you do the sampling, you have the barcode scanner. So you put a label there with the scan. So starting from the sample entry into the lab to the destruction, it goes to the automated process of scanning. The advantage is that anytime the lab manager allocates a sample to the analyst, he simply allocates in the system, and on the sample level, you have the barcode. So you scan it, and then automatically get tags. The advantage is that in that process, you can never take a sample which is a different lot number or a different product. Because what happens is that if you try to take it, or somebody by mistake takes a different sample, it doesn't get integrated. So please try to, it's a very simple process like you do in a supermarket. But many of the organizations, I see that even today, the sample management is still a manual job. So the whole process, actually, you can automate it. Then schedule management, through the paperless lab, all the solutions which are available, you can very well schedule your sample planning, which HPLC or which chromatographic system is available to you to work on. So you can do a very good schedule management on that. Inventory management, sometimes at the last moment you find that there is one column which is not available, or the working standard which is expired. So the whole process, the system gives you an indication, gives you a pop-up that, hey, in 1 month's time, this working standard is going to expire. So you can very well control your inventory management. Analysis and data management, of course, there's a core of the laboratory activity. Then you have training. So many times, if you have got a very big lab, like you have got 200, 250 scientists there working, so all of them needs to be trained. All of them needs to be qualified. It's a humongous job to monitor that manually, and then to keep a track who is trained on what and who is qualified in which test. So the system, you should automate in that way that the sample, even when I try to analyze, I cannot do that if I am not qualified or if I am not trained on that particular recipe. The system says, hey, you are not qualified, you cannot start the analysis. Then investigation, CAPA, change management. There's a buzzword of this GMP process now. All of them you can automate there. Then the hardware. There's the intrinsic part of the system. Now, how you can do that? Which system actually takes you in the journey? You have the lab there. So the LIMS, which will take care of So put everything in the LIMS, starting with samples. The moment you take the sample, it goes into the LIMS system, starting from the sample to the release, everything can be controlled to the LIMS. You have the SAP, because many of the companies, SAP is a very well-developed software. So many of the companies will be SAP. So integrate with the SAP management. Then you have got quality assurance management system. Many of the companies have implemented that, all this deviation, OS. So all these things can be linked into that LIMS. Then as I already told, the importance of barcode. Then document management system. You have got now the process in any company is very, very complicated. You have got 50, 60 customers, 100 customers. Everybody, if you're running an API plant, every customer will be having some kind of a different PSD based on their formulation requirement. So many of the times if you try to do that manually, control that, you have a problem. So do the whole thing automated in the system. The DMS system will tell you which specification, which test procedure will be having, which kind of controls, and then allocate to that customer to that item code. So you'll never land up into a wrong batch release to some different customer. Then LMS, there's a learning management system. So integrated that with the LIMS. So it will always tell you that this person is straight, this person is not. So the whole training can be automated to that. And then obviously the core function is the database. Advantages, all of us know that So the highest level of compliance, reduction in human error, that's one of the big problem now with the FDA compliance. You have got so many OSs out of specification, out of trend because of the human error. Analyst comfort. What I always say that we have all the qualified, very, very qualified people. They are educated people. Sometimes they are postgraduates. And then most of the time I find that in a manual process, they actually spend their time in a very unproductive manner, keeping a trap, which format to fill, there shouldn't be any mistakes. Instead of that, through automation, you can actually use their time for the more productive job. The data analysis, what kind of analysis they are doing, what kind of problems they are facing, what kind of trend they are getting it. So those things actually, you can concentrate more, you have the whole process automated. Enhanced productivity, of course you get it. Reduction in failures, ease and traceability of data. Anytime you can archive it, anytime you can go through the data. Confidence and quality of the reports, you don't have any mistakes in the certificate of analysis. Somebody doesn't have to again go through, check whether you have generated the correct report or not. Then get into the hybrid system. This is one of the area I have traveled to many, many companies in the world. I always don't find a logic of it. For example, if your chromatographic systems and data, currently, most of the companies are having their online data system. I still find people getting the chromatograms printed. Please get rid of all the unproductive work and the hybrid system of manual, computerized. If there is a good traceability, if there is a control in the computer system, I don't find why you should again print the chromatograms. Only thing you should be having it, a good traceable system so that during the audits or all, you can easily, immediately retrieve the data. So get rid of as much as hybrid system as possible. Now, challenges. This is the place, this is over the 25 years I have faced that these are the most important challenges. One is that instrument software hardware integration. When you decide today that I want to go down the line 5 years from where I stand, the important part is that you have many, many instruments, software, hardware you have bought over the years. In a company, you cannot just make all of them redundant today and then start afresh. You cannot do that simply. So the integration part is a very, very important thing with the existing instruments, existing software, and then taking it forward. Project management, very core team. Always decide on a roadmap. What I'm going to achieve in next 1 year, what I'm going to have it 5 years. If you want to do everything together, not possible. Have a good project management team, have the vision that 5 years down the line, I want to be here, and then you decide on your systems. Otherwise, sometimes you go on buying software systems, and then after 1 year, 2 years, you find something is not getting integrated to the other, and then you have challenges. So everything is to be rewritten then. So don't do that. Vendor dependence and troubleshoot. you need to have good infrastructure within your company. You cannot simply depend all the time on the vendor and the troubleshoot. You will have to build that skill along with the vendor, that small problems I can resolve it myself. Otherwise, when we go for a paperless journey, the thing is we are not all of them are experts in the IT solutions. So what happens every time during the world, even for small things, you are struck up. And then the music starts. There is an absolute kind of a disturbance occurs. So have a good infrastructure so that you don't have to depend on the vendor all the time. Customization. Customer is the first. So what happens in the process is that when a vendor approaches you, and most of the time I find that we try to photocopy of the existing system into the automated system. That's the worst thing. You cannot really expect that everything today, whatever is there in your manual system, that will be photocopied into the automated system. And then what happens, you try to tell them, hey, we need this. The vendor cannot every time tell you, we cannot do this. Because then you find that the vendor is not ready to help you. So it's a catch-22 situation where we try to do customization. So my request is that first try to assess the vendor, do customization to the least possible. Because if you do more customization, then the problem is you land up into a problem. One is the project delivery time. Second is that the vendor has to configure a lot of things. And that lot of configuration will ultimately give you trouble in the output. Mindset, very important. Please fix your mindset where you want to go. In the pharma industry, unlike many other industries, we are still not automated many of the process. So mindset is extremely important. Financial aspect, cost is always important. So try to figure it out what actually suits best to you. Proactive management. Don't think today if you are bringing a software in the lab, don't think for today. Think of 5 years, 10 years down the line. Must to have. To me, if I design a paperless lab, this will be must to have for me. First, you do the assessment in a very sound manner. See that what suits you best. And in the process, you should select the vendor. Many of the times, just based on some kind of initial assessment, you go on buying the software, and then you land up into a problem. Robust IT infrastructure. See the bandwidth of your IT from today onwards. If you make a road plan for the next 5 years, see what kind of bandwidth you'll have to have, what kind of server you'll have to have, make that road plan. Skilled workforce, the robust IT infrastructure, then the skilled workforce, as I already told you. Just not to have paper and glass. Just don't, as I said, don't make a photocopy of the existing system into the software system. As it is, customers only when necessary, training and troubleshoot, and time-bound implementation. Time is very important to implement all the projects. Otherwise, you are not ahead of the time. So some of the features, you should always have it. Even now, I find in companies that we have very good chromatographic online integration systems. But still, I find some of the places, these are not done. So online calculation, you should always have it. Auto integration, you should not depend on the manual integration. And FDA always have a doubtful mind once you say that I am doing manual integration. Electronic submission sequence, you just electronically submit, get an electronic signature, no printing of chromatogram. The titematic analysis, I've just tried to figure out that what are the important analysis you have. I mean, important in the sense that different types of. So similar way, you have got auto titration, get the auto weight transferred into the system, no need of writing down 2.5 mg, and one day you find it that you have suddenly put, instead of the decimal, you have put 25 mg. An error, but you will land up into an OS result. Electronic submission, and then review with the electronic signature. Other instruments, so try to have all these integrations, balance, pH meter, viscometer, density meter, all of them, you integrate it. Real-time data featured in the test data sheet. There is no need to have test data sheet manually printed. You just do everything online. The test data sheet will be fetched into the computer system. No manual. Record management, as you know, I find that many times, because CFR requires that you should be having a log of the instrument. build up in the system that if you have an automated process, all the logs, whether it's column, whether it's standard, whether it's the instrument usage, everything in the system. No need for reconciliation of the working standard in a manual way. Have that reconciliation build up in the system, online system itself. Then you have the authorization, real-time usage entry, retrieval of the records, and e-test data sheet. Features data management, you know, audit trail is very important. This is a very important thing, the exception report. In a big lab, you have around 50, 60 pages of stage data sheet. And the reviewer, if you tell them that you have to review it, it's a nightmare. You can miss out. So have a system, when you are going for the paperless lab, there should be an exception report. For example, one day, you don't have much of the quantity of the standard. For example, you are supposed to have 25 mg, you have 30 mg to 100 ml. You don't have that. So you want to take a deviation. So you want to do 15 mg in 50 ml. So the final concentration is not going to change. So I keep aside the robustness of the method and all, but just for taking it as an example, your final concentration is same. So you don't have any problem in using that. So first of all, when you try to put the system, if you take 15 mg weight, it goes into the system, it should not allow you because it's written that the method that 30 mg to be taken. So the system should automatically boot you to your quality assurance manager system that, hey, you need a deviation. So the deviation is locked, the impact analysis is done, then you allow. In the final report, when the report comes up for the review, you should have those exceptions only where you deviated from the process. The easier thing for a reviewer then is that he or she doesn't have to go to all 25, 30 pages. He or she has to concentrate only on the exception report, decide it for the release of the batch. Epitome of paperless lab. What you try to do is that integration of maximum lab instruments, auto-weight transfer, dilution is still a big problem in the sample analysis. Try to automate that process. There are instruments available where they do auto-dilution. Only to qualified, you have, in a big lab, you have got 70, 80, 100 HPLCs. This calibration to be done in a periodic manner. The qualification is to be done. Always build the check factor in the system that somebody should not be able to use an unqualified or non-calibrated instrument. The moment you try to use that, the system will give a pop-up that, hey, you cannot use this because it is still under calibration. As I told you, the training completion already, if you link it to the system, auto sample management through scanning, I explained it to you. Not only the auto sample, even the volumetric solution, the working standard, column, everything, you scan it, just build it in the system that it goes directly to the limbs. You don't need, even for volumetric solutions and all the working standard, to manually write. You scan it, system goes. And the system will be having the control that, let's say for X product, this is the current available working standard. It links. If it is not there, it doesn't link. So the control is always built up. And then remote access data with the real-time monitoring, and then statistical analysis. In this kind of a scenario, your status, there are so many statistical kind of data, actually, data analysis you can do. So this is just some of the things that, chemical standard column, what kind of things you can manage through the automated system. It actually gives you that restriction. You cannot use an expired standard. You cannot use an expired column. Notification gives you, if you build them in the system, it gives you 1 month before the expiry. Hey, these are the chemicals, these are the working standard are going to expire in the lab. So start procurement from now. Auto purchase request. If you build up that 1 month before, this particular PO should go for this much quantity. If you define that this is the minimum order quantity, system automatically generates a purchase order for that particular material, and it goes to the purchase, and they can release the order without any kind of discussion, authorization, and all. Sample management, as I've already told you, scheduling, planning, result recording with auto calculation. This is some of the things currently have come up that you have, and these are real instruments. The auto dilution part, you can totally automate it. So you take the weight of the sample, let's say 25 mg, you put a volumetric flux there, the weight goes into the system, then you define the dilutions there in the system, and then system will automatically capture the dilution and take the required dilutions there. Dissolution apparatus, we have now robotic dissolution apparatus, then it is integrated with the UV, then PAT tools are, people are randomly, not randomly, people have started using it, the PAT tools, and then building of artificial intelligence, which we are going to talk about in the other speakers during the course of the day. This is another area which is always a concern for me, is that microbiology laboratory, as if all of you know, for a sterile manufacturing plant is extremely important. Just imagine a situation in a sterile manufacturing plant, on a single day, you have got 200, 300 of plates, microbial count plates, it is exposed in the area. Just imagine a situation under a grade A, the sample location gets changed, and you have one day an OS result. What happens? You get an absolute different result than that particular location. It can become a catastrophic issue in the company. And when you are dealing with 200, 300 plates and everything manual, it's very difficult. In the chemical lab, chromatography and all, you have some printout left. You do an analysis, you have a chromatograph. In micro lab, today you are taking the count as 10. Down the line, 1 month later, nobody can prove that it was 10 or it was 1. That's the kind of microbiology, the risk you have. And today's solutions are available. I'll show you from the real life screen prints where the systems are available, starting from the receipt of the media up to the analysis. The whole process can be automated. And these are the realistic solutions available, and people have started implementing those. Now, if you really look into, I have divided the core micro activities into environmental monitoring, personal monitoring, water analysis, media management, bioball, the culture management, MLT, that's the testing part, and the organism identification. These are overall broad-based, the micro activities. Here I have given, I don't have much time to really go through all of them, but this is the way you can actually build up in the system that the environmental monitoring, you can create the schedule, the environmental monitoring plan, it gives a pop-up on daily basis, what sample, where to do the testing. Then receiving the report, the environmental microbeam plates in the lab, then media, you registered the media and use the media stock, then preparation, sterilization, qualification, instrument integration, weighing balance, pH meter, these are all you can integrate. So the media, which you're going to weigh, which you're going to pH adjustment, this goes directly into the system. Bible culture, register of various types of Bible, these all you can actually fit it into the system itself. Water analysis, again it will give you a monitoring schedule, the collection of samples, except the sample collection manually. Everything can be done in the system itself. Then sterility testing, organism identification. Now these are real life, real system photographs of the screenshot I have taken. So what happens that the micro activity starts in the lab from the media receipt. So the moment you have a media receipt built down in the system, let's say you want to use the SCDA for this kind of analysis. So the SCDA media, you register that these are my approved vendor, these are my the requirement from that media. So system, the moment you receive a lot, the system has got the inbuilt authorization where it only accepts what is fed into the system vendor with respect to lots. If you say that I don't want to accept a media from a vendor which is not having at least the 6 months of expiry period left. So build that in the system, and system will not allow you to take any material vendor gives you which is having a less than 6 months expiry. So then you generate this kind of media level. And then the barcode, the scan, you do that scanning, and then it's in the system, and throughout your life cycle, you have the traceability. This media has been used where? This is a sample of prepared media. This is a sample of environmental monitoring plates. This is a, this is a very important thing, environment monitoring group level. Group level means you have a grade A area. You'll have to put around 10 plates. Now, in a grade A area, you cannot take the plate on the back of it a manual level. It's just not allowed because of the microbial control you have. So what do? You select 10 plates I'm going to take from a batch. You allocate in the system. And then what you do, outside the cubicle, outside the manufacturing suite, put a level like that, that whatever 10 plates go inside, you scan just outside that room. The moment you scan this one, that means these 10 plates are allocated to this particular area at a time. So means these 10 plates are going into that area, and the whole system then takes into for reconciliation accordingly. Now, these are real-life plates. So what we do is that you take the environmental monitoring plate, have a scan barcode, and then the location where you take it, the location will also, where you want to place the plate, there is also a barcode. So the moment you take it there, you integrate that. The moment you integrate, no other plate can be actually integrated with any other location because it is one-to-one. So the biggest advantage is that you can never end up into a plate missing or a plate giving it to location which is not that particular location. Now, this is a very interesting thing. You know, it is very well possible that what is the next step? You take a plate, then go into the incubator for incubation. Now, on a single day, you will find in your incubators more than 100, 200 plates of different days lying. And the complication is that the plates will have to be exposed in 2 different incubator conditions. So after some day, you'll have to take same plate out, go to a different incubator. Just imagine you take a plate which has not yet completed the incubation in a particular incubator. So actually, the microorganisms will not be detected. So how do you automate that? So what you do, your plate is having the barcode. You put the same barcode here in the incubator, and the incubator will be having the conditions. So that is designed in the system. So you take the plate, come to the incubator, you scan it. That scanning time is the start of incubation. Now, let's say somebody by mistake takes it out 1 day before, by mistake. The system does not allow you now. It says incubation time not completed, cannot end the incubation. So you cannot take a plate which has not completed incubation there. That's a great control I'm talking about. Then the automation activity. You know, these are the tablets which are used for controlling all the systems. There is no paper is used. And this also used as a scanner. It is, at the front of it is a scanning mechanism. So every time the microbiologist go to the floor, go to the lab, he doesn't have to carry anything except this tab. He takes the tab, build down in the system, and then scan it. His job is done. So instrument integration, so weighing and pH and all, you don't have to manually again type it. System, the moment you weigh it goes into the system that this much weight and pH has been recorded. Now, just a screenshot of some of the things. No need of manual test data sheet. Everything you automate, be in the system. Review checklist, logbooks, and reports. Review is an important mechanism in micro. So you don't have to do that review manually. You can do that in the system. Now, just an example. It shows in yellow, means incubator started. Incubation completed, it's green. Unqualified media, you cannot use it. It will show you no. Similarly, it will also show you which one is review pending, which is review completed. So everything is automated. You just don't have to worry about anything. Checklist for review. The reviewer can have a checklist in the system that I want to review this parameter before release of the batch. So he has to come to the system, check those online into the microsystem, and then he just put, that these are the checklists, I have done it. So it has a time and name stamp. So immediately you know who has reviewed at what time point, and these are all green, means the system is reviewed. The excursions. Anytime you have an excursion, you can fix it, whether it's an out of train or it's an out of specification. Systems gives you an indication that, hey, red, you are not in compliance. This is with respect to pH. You have defined a pH limit of 7.1 to 7.5. Your pH is 7.7. It gives you an indication, look, it is not compliance. It doesn't even accept it. You cannot approve that pH to take it forward. It will never going to accept it. Contemporaneous observation, recording, and verification by second person. It is very important that many of the times the inspectors are expecting that if there is a micro data reviewed, the count is reviewed by one person, it should be contemporaneously checked by another person so that you don't have a mistake. Now in the system, you can do that contemporaneously by one person by counting it, another person by checking it. This is an important evidence. What you can do here is that, after the count, the plates are destroyed. You never have any evidence. So in the system, the instruments are now available, which counts and then automatically takes a photograph. That photograph, it's a real life. It's not something is built up. It's a real plate where you have got the barcode. The system takes the printout and it then archived into the system. So any point of time, 5 years back, that particular day, that particular sample had 5 count. So during the audit, it becomes very easy. Hey, I recorded 5, there are 5. If you want, you can count on the photograph itself. As I told you, these are some of the scanning we can do to the location and the media plates. Then the more control activities. You cannot use an out of calibration under breakdown instrument, no manual test data sheet. Final release, again, based on the exception report. The exception report will say, this is the OT. Even if you have got an out of train, the system actually tells you that please log OT in the system, otherwise I will not allow you to release the batch. And then any expired, unqualified media, you cannot use it if you have this control. This is again a suit kind of a concept in a sterile unit. If you have this kind of a suit, and any point of time you build up in your system, at any given point, you can really know. If you just click on that particular locations, it gives you the status. That when it was due for your monitoring, when it is overdue or not, whether you are in compliance or not. This also, anytime you go to the suit, click on that button, and you get the train. You want 5 years train, 1 year train, last 25 days train, you just get in a minute. You can get all the trains. It becomes very easier for you to see all this. And this is different, many of the train reports you get it. So at the end, I know I have gone through very fast because it's only 20 minutes, but I have tried to give an overview that today, you can automate everything. And solutions are actually available. So it's no longer a dream. In fact, it's a necessity. Thank you very much.
Thank you very much, Mr. Bose. For a very detailed presentation, he has a lot of things to share. And I'm sure all of you have more experience, but a humble request for all speakers, please stick to the time, okay? And message well taken that paperless dream is a necessity, okay? So I'm sure you have some questions, but due to time limit, we will do the question and answer only after the first session. So please hold on with your questions for 2 more talks. So going to the next talk, drug discovery is a long process, and it takes 10 to 12 years or sometimes more than that. And in addition, only 14% is the success rate currently. So bringing the new drug to the market is a big challenge in pharma industry today, and lot of efforts in being taken to reduce the time. Regulation, of course, is one of the reasons to delay, but it is important for patient safety and efficacy. And that's the reason now artificial intelligence and machine learning are said to fasten this process much faster. And today, we have Dr. Jimmy Yen, CEO of In Silico Medicines, and he is also working as R&D and business development, and he will share how AI and machine learning can accelerate the drug discovery processes. Please welcome Dr. Jimmy Yen Chu Lin.
Hello, can you hear me well? Yes, okay. Good morning, everyone. It's on behalf of In Silico Medicine, I would like to thank Shimadzu to invite us to share our visions in transforming the future of pharma. So my name is Jimmy, so I'm based in Taiwan. So actually I worked in Singapore for several years, so it's so nice to come back. And actually it's because my first child was born here, so here is just like my second home. Okay. So today I'm going to talk about our company like using AI for drug discovery, especially in identify the new targets, followed by using the AI for molecular generation. We also have some efforts in prediction the clinical trial outcomes, but today I'm not going to talk about this part. So I think everyone is very familiar with these figures. So nowadays, like we have a very high rate of failure rate for developing into, drugs from the bench to the clinics. And overall, because of this very high failure rate, so we need to spend more than $2 billion to develop one drug into clinics. So that's why we are thinking like if we can use AI to really facilitate the process and reduce the cost and reduce the time. So this figure is adapted from the Nature Review Drug Discovery in 2010. So this is the figures summarized by Eli Lilly. Actually, they summarize how much they need to pay, they to spend, and how long does it take for put one drug from targets to hit, and then further way all the way down to clinical trial, and at the last stage, the launch in the clinic. So overall, it takes around like more than 10 years, and in their calculation, they need to spend like around $1 billion, $1.7 to put one drug into clinics. So that's why you can imagine that nowadays the drug discovery process is kind of a long journey, and then you need to spend time and spend lots of money into that. So these figures is from Deep Knowledge Analytics for the pharma division. So actually, many people think that probably the AI can really help the drug discovery process. So in this report, they separate the company in different segments, like using different AI technology to solve the different parts of drug discovery problems. So in our company, In Silico Medicine here, so we are focusing on doing the drug design. And there are some companies, probably you are very familiar with this, like doing some other clinical trial designs or using their in-house algorithm to doing the repurposing existing drugs. But I have to say that there are so many companies here, but some of them are really good, like us, so that's why we are here. So, and some of them are just starting their business, but we still need time to prove that they can really bring an impact to this in this industry. So our company was founded in 2014 from Rockville in Johns Hopkins universities, and during these years we have global footprints in eight different countries and regions, and so I'm in charge in Taiwan office right now. So totally we have 70 people around the world, and half of them are computational scientists developing the deep learning algorithm for drug discovery, and half of them are medicinal chemists, biologists, working closely together with computational scientists to design a good algorithm for drug discovery. So because of our recent efforts, we are recognized by several practice awards, like Frost & Sullivan Best Practice Award 2018. And last year, we were nominated by CB Insights as a top 100 AI companies all over the world. So as I mentioned, this is a long journey in drug discovery process. So In Silico Medicine is aiming for using AI to shorten the process. So we mainly use AI for preclinical stage in our target identifications and molecular generations. And we also have some efforts in designing machine learning models for clinical trials. So we have three pillars. The first the first platform we have is called Pandomics. So we use AI method to identify new targets from the patient's omics data. And once we have targets in mind, we use algorithm to generate new molecules. And we have another effort in building a deep learning algorithm to predict the drugs outcomes in clinical trials. And this one, we are currently working closely with a big pharma. So I cannot share too many details today. So the first part, I will talk about how do we use AI or bioinformatics method to identify new targets from the omics data. So we have several proof of concept papers, one of them in Nature Communications, and then I cannot go to the details today, but I will also show you one examples that we collaborate with Johns Hopkins Universities. That one was also published on Nature Communications, that using our platform to solve their problems. So our platforms for target identification is called Pandomics. So it's abbreviation of Panda and Omics and Pandomics. So we mainly works on omics data, transcriptomic data, most of the time. And then we have an internal algorithm called iPanda, can give you a very confident calculations of pathways difference between the patient samples and health people samples. And then once we have some signal pathways in mind, so we can use our in-house algorithm to identify new targets from these data. And then in this platform, we also integrate almost all the world available database to incorporate into this system, so to make into a user-friendly platforms. So in these systems, we collect around 1.9 trillion data points from all over database, plus our internal database from our collaborators or from our internal research. So we incorporate geo database for omics data. We collect Campbell's for small molecule data. We also have some data from US Patents Office. So once you have the omics data, you upload it to the system will calculate the signaling pathway difference between two samples, and then we will identify the targets for you. So here is a glance from our platforms, like the target is suggested by platforms. So in our platforms, we are not only using the omics data only, we also do some data mining or data mining approach, trying to see the targets we identify by our algorithm is valid or not. By using some data mining approach, we search all the clinical trial data, funding information, patent information, and also the publication information, trying to see if any of these new targets suggested by our systems has any association with the disease we are interested in before. And then we will give you a combination scoring here, and then can give you and prioritize the top 20 targets for the users. And so the user can easily decide which targets are you going to pick up and do the further validations. So here give you an examples that we have with the Johns Hopkins Universities published in Nature Communication last year. So here we help them to analyze some real patient samples from the TCGA database. So the questions we want to ask here is why some patients are not very respond to the immune checkpoint inhibitors. One hypothesis is that because of TGA beta pathway over expressions, and so it makes the T cells inhibiting the T cells functions. So here we using our iPanda algorithm to analyze the real breast cancer patient samples from TCGA and clearly show that TGA beta is highly overexpressions and it has highly correlation with FOXP3, which is the T regulatory cells biomarkers. So based on this finding, they designed a new generation of antibody. In a variable region, they still keep the CTLA-4 or PD-1, very famous checkpoint inhibitors. But in a constant region, they put an extra TGA beta receptor active domain to quench the TGA beta level in the tumor microenvironment. So based on these results, in this paper, they did a lot of study in vitro, in vivo to show this new generation of antibody is better than the CTLA-4 or PD-1 or even a combination of these two antibodies in some xenograft models. So based on these results, they spin off a new company to promote this antibody further to the clinics. So this is the perfect showcase to show that how do we use our platform to help your drug discovery problems and then to really shorten your process and to make it into clinic as soon as possible. So once you have targets in mind, or you already have targets in mind, so you can use our algorithm our platform to generate new molecules for you. So we have a series of papers on the PubMed, in the PubMed, so like you can search, you can Google it, and including some proof of concept papers in designing new deep learning models, and also some proof of concept showing case, synthesize the molecules and then test it in the in vitro and in vivo assays. So recently we just accepted a paper on Nature Biotech, which I will briefly introduce later, to show how do we use our AI platform to generate the molecules, and then the molecule can have really show some efficacies. So as I mentioned earlier, nowadays the drug discovery process is not very efficient. It's just like looking for a needle in a haystack. In Chinese, we call it 大海捞针. So how do we really shorten the process and to make it faster? So we leverage the two very famous deep learning algorithms in the world. Like one is GANs, generative adversarial networks. One is reinforcement learning. So trying to leverage the power of these two algorithms and to make it, and then to use it for molecular generations. So the GANs, generative adversarial networks, was first developed in 2014, and at the very beginning it was used for image generations. So in the GANs, you have two networks. One is generator, discriminators. In discriminator, you will use the real sample to train the discriminators. Let's say you want to generate some flower pictures, so we will use the real flower picture to generate the networks. So discriminator will know what is the real flower pictures, and the generators, you just train it how to generate the pictures in general. So at the very beginning, the generator will try to generate some flower pictures, but discriminator can easily tell that these pictures are fake, it's not from the real data, because the generator doesn't know how to generate a real flower pictures. So it will be the iterative cycles. So at the end, generator will try to generate a perfect image that the discriminator cannot distinguish it at all, if it is from generator or real data. So here are some image that generated from GANs algorithms. So you will see they are pretty much the true face, but actually they are not exist in the world. So they are the fake image generated from the GANs algorithm. So we are thinking that if we can use these GANs to generate some molecules. So let's say if you have a non-active molecules with certain scaffolds, can you use some GANs algorithm to do some scaffold, to design a new scaffold molecules and with better activity? Because I think the chemists estimate that the whole chemical space that can be synthesized and the chemicals with drug-like properties around the 10 to the power 16. So this number actually is more than all the stars in the universe. So you can imagine that all the possibility here is that you can synthesize, the molecule can be synthesized with drug-like property is huge, it's enormous. But nowadays, when we do some high-throughput screening or even the DNA encode library, trying to do some virtual screening or do some real screening to find the perfect molecules, I think the library size is around 10 to the power 10 or even 10 to the power 11. So it's really far beyond the whole chemical space you can explore. So we are thinking that if we can use AI to really explore the whole chemical space more efficiently. So recently we have a Nature Biotech paper just accepted 1 month ago. So this is the exercise we have with Wuxi Aptex. It's also our key investors. So before they invest us, they want to challenge us if we have capability to design some molecule in a very short period of time. So here, the case we are showing here is a DDR1 kinase. So Wuxi Aptex only provide us the names of kinase. So our team spent 1 week to collect all the kinase inhibitors in the publicly available database, and then use this data to train our models. And then the model will start to learn the features of these data and generate the models, generate some new molecules. with desired properties. And so it comes to 30K structures after 12 days. And then we use some computational AI drug design approach to narrow down this compound down to the 40. And then we discuss these 40 compounds with the chemists in the Wuxi, and we prioritize 6 compounds. And since Wuxi helped us to do the synthesis and do the biological test, it turns out that 2 compounds show very good activity, and then also with very good IP positions. So in total, the whole exercise only takes about 46 days. In the 46 days, we can generate a perfect, I don't want to say perfect, but can generate a very good hit molecule, even the lead molecules, in a short period of time. So this is the model we use in our papers. I don't want to go to the details, but you can read the papers if you are interested. So basically, this is the deep learning model we use. We call it VAE, conjugate with a reinforcement learning strategies, trying to optimize the latent space here to teach the generators to generate a perfect molecules that with our desired properties. So here we want to design the kinase inhibitor and selective inhibitor targeting the DDR1 kinase. So this is the models we developed 2 years ago. So and then we take another 1 year to get published on Nature Biotech because we spend lots of efforts in convincing the reviewer that these models really works. So nowadays actually we have more complex deep learning models in-house to trying to tackle different drug discovery problems. So not only for the kinase, we can also work on some difficult targets like GPCR or protein-protein interactions or some mutant form kinase inhibitors. So at the end, actually, what's the output from our AI-based drug discovery platforms? Actually, we want to generate a patent-free molecules with very good novelty and then with good diversity, so we can discover some privileged scaffolds, so the chemist or the AI algorithm can do the optimizations afterwards. So also the final goal, the ultimate goal for us is want to generate a good drug-like molecules. Like we don't really need to find the needles in the hat stack. We can directly generate some perfect needles directly. So this is our ultimate goals, and we are on the right track to this goal. So at the end, so actually we are the only company in the world like have an end-to-end AI platform for drug discovery, from the target identification, molecular generations. So doing the molecular generation, we also can put some the properties we want into these molecules, like good ADMET properties and good efficacies, for examples. And we also have some efforts in predicting the clinical trial outcomes. And there's a recent news from us. So actually, we just closed a B round, which we raised $37 million USD. So our leading investors are mainly from China and also from some investors from Silicon Valley as well. And recently, we have a big deal with Pharma in China, City Farm. So we have 2 projects with Slan. Its total value will be $200 million USD. So in these projects, we are working on very difficult targets. It's not it's very difficult targets like with very limited information, but we will try to use our AI to help them to do the drug discovery problems. So I think I hope that in my 20 minutes talk, I can give you a brief introduction about what we are doing and what we can do. And then from my point of view, I think the AI for drug discovery is still in its infant stage. So we will see if it will make a great impact to the drug discovery industry in the very near future. Thanks for your listening.
Thank you very much for that really interesting talk. And I just realized that AI can not only reduce the drug discovery time, also reduces the presentation time. Okay. So thanks for that. Moving on, big data and data analysis is extremely important and useful for better understanding of process as well as to ensure the quality. Hence, it is important to generate lot of data, but more importantly, rightly analyzed and managed. The quality aspects in pharma is ever changing. And in the next and final talk of this session, Dr. Kavita Vengurlekar Head of Data Science from Caliber, is going to explain how data analysis can help to improve the quality and the process. Dr. Kavita, please welcome.
Thank you, Mr. Indrajit and Dr. Jimmy, for setting the stage for me. And thanks, Shimadzu team, for inviting me. I'm audible now? Let me begin my session by sharing one incident. Today in the morning, I was catching up with my daughter, who is aspiring to become a lawyer. And in midst of the conversation, I just commented to her that she does a lot of analysis. And she said, analysis is a cool thing. Only difference is that she does for facts and evidences, and I do on data to get more facts and evidences. And I said, wow. Good morning, ladies and gentlemen. I'm from India. My name is Kavita, and I represent Caliber Technologies Private Limited. Till date, my roles and responsibilities are to integrate data, manage data, process the data, transform data, and of course, analyze data. And hence, data, databases, data science, and writing algorithms, they are my bread, butter, and of course, a jam as well. Before dealing with pharma data, I dealt extensively with automotive sector, as well as banking and financial sector. To have a data-driven organization, there are two needs. One is a business need, and second is a regulatory needs. APQR, Annual Product Quality Report, and quality metrics, from the regulatory needs, they are the big ticket items. And there is also a third need, which I am going to reveal at the end of the session. Let's quickly go through the agenda so as to know how my session is organized. First, we will see how suddenly the data has become big. Second, now when the data is big, what is the roadmap and architecture to harness that data or information to convert that into actionable insights? And the third, which is very, very important, that is a possibilities and takeaway, that is nothing but a use cases. Sorry. Way back in 2009 and 2010, in IBM Research Laboratory, we were being asked to do a POC immediately on a big data. That time, big data was defined and characterized by 3 V's. That is, data volume, data velocity, and data variety. With boom in internet technology, we were anticipating data velocity, and hence, the computer giants, they were gearing up to tackle it. For the data volume, Yahoo scientist, he has introduced Hadoop file for the database storage system. And till that time, we were storing just petabytes of a data. Because of the introduction of the Hadoop file, we were able to store exa and zettabytes of a data, and that actually changed the entire game in a data storage, and hence today the data storage is a commodity. Let's talk about the data variety. Data variety was there and it will be there. When I say what is a data variety, for me, maybe a disparate sources or a silos of a data or a data island. Let us have a look in the current state of a pharma industry, what it is. In a lab, we have a beautiful system which is called as Laboratory Information Management System, and it does fantastic job. But the data is stored and intact in its own database. So the same story goes for QA, same story goes for the batch and manufacturing record, same story goes for the training. thing. And believe me, with my experience, I must tell you, pharma industry is not an exception at all. Other industries, they also face the same challenge. The only difference is they actually anticipated the ripple effects of these data islands. Because of that, they were ready back then in 2000 and 2010, and they have taken a corrective action. Pharma industry, I must say, is little behind. Now, what is the ripple effect of having a data island? First of all, let us see that. I will be mentioning only 3 ripple effects, and this is not an exhaustive list, but just to give you a glance. First constraint is interdepartmental analytics. That is a constraint, because lab data doesn't talk to QA data doesn't talk to ERP data. And hence, even if we would like to have interdepartmental analytics, there will be a manual consolidation. Time and resources will be engaged and blocked for a certain period of a time, and probability of having an error because it's a human is more. Let us see the second ripple effect. The classic example, annual product quality report, where regulators nowadays, they want comprehensive report from all these data islands. Same story, here again repeats. Manual consolidation, time and resources are getting engaged, and of course, the probability of having a human error is high, and I know that this error in APQR will cost us more. The third ripple effect is quality metrics. This quality metrics has not hit us yet. Let me give you just a brief about quality metrics. In the year 2013, FDA started this quality metrics initiative with a mandated program. Industry started pushing back, and because of that, they changed their mandated program to the guideline. ISPE in 2014, they launched a pilot project called as Wave 1, wherein they said, okay, we would invite only the volunteer companies or the volunteer pharma organization to execute only the first top 3 reports, not the entire report. Here, ISP is very, very clever, because they know today FDA has made this quality metrics as a guideline. Tomorrow, it is going to be rule. Now, when the company started doing a work on this, they see this beautiful web design. Again, the same story, right? So, what is the solution? How do we approach to mitigate this problem of data island? The solution is data integration. What does that mean? That means we still continue to have our transactional database management system continued as is. We don't have to worry about it, but only we have to build a top layer on it, which will extract and ingest all these islands of a data into one big database. Now here, the technology can be enterprise database, can be data warehouse, can be data lake. Now, let us see what is the advantage of that. First, all these ripple effects will be handled. Second, data integration is the prerequisite for the data-driven organization. Third, I understand that, I missed that point, that pharma industries, they are still continuing to maintain very, very important information in their Excel sheet. Now, having the data integration in place, these Excel sheet will come into the electronic system and will be attached to the audit trail. Another important use case for having a data integration in place is if we want to have some information of lab with reference to QA, that is a correlation, this correlation matrix analytics is possible because data integration is in place. Right. There is a new metaphor for data nowadays. Do you know what is that for data? Anyone? No? Okay. data is new oil. That is a metaphor. I understand data cannot be gold, data cannot be silver, but data is a new oil. But there is a context behind this metaphor. As long as data is unrefined, it is of no use. The moment we start doing data analytics and data mining on it, it creates exceptionally and very beautiful insights out of it. Also, there is a historical context. I'll just tell you one story behind it, which our statistical professor, they used to tell us. Dr. Jimmy will agree with me. There was a statistician in Greek. His name was Thales, and he was a mathematician as well as statistician. And this is in the year 500 BC. Kindly note, 500 BC. That time, he has predicted with a 50 data set point how would be the weather. And from this weather, he predicted that harvest for the olive would be best. What he did, he well in advance, he rented out all the olive presses mills, and he achieved riches. So that was the historical context to it. Now, let us quickly see what are the key ingredients for data-driven organization. Of course, data is the main key ingredient. Strategy is important. People, we have to add little bit more data analyst as well as data scientist. Technology, ample out in the market. Culture, yes, we need to change. In the interest of a time, I'm not going to cover all these topics, but I will be focusing more on data. Now, so what we do, there are technologies which is available in the market, and those are called as ETL, extraction, transformation, and load. With these ETL tools, they have a capability of ingesting disparate sources, collecting it, storing it, and consolidating in the base of our pyramid. Hence, interdepartmental analytics, the first ripple effect is being addressed. On top of it, another layer we create that is called as data mart or data warehouse or data lake, whatever the technology you choose. And APQR, multi-site metrics report, process capability, quality matrix, statistical report, CPK and PPK, they are also being addressed by this third layer. Now, to complete my pyramid or a data pyramid, when a base of a pyramid is completely filled up with clean, trusted, and integrated data, we have to just keep one data science layer on it so that insights are getting extracted. Again, for data science, lot of technologies available in the market. Whatever you want to choose, you can choose. Now, these insights, they are going to give us the inside story of our system. On that basis, there will be some changes which we would like to have that in our 4 M's, right? That is man, machine, method. And again, my model would be deployed, and again, another insights would be there. So this cycle is continued, which is making our system robust day by day. Now, in the year 2004 and 2005, my team and myself, we were working to integrate data for General Motors across 3 continents. And we were responsible not to integrate their data, but to analyze it, to give the statistical report, and as well as we have to give the dashboards as well. And these reports, they were going directly to the GM higher management. In one of the meetings, one of the top boss, he said, this team in 3 months knows about each and everything what he knows in 45 years. So that is the power of this data pyramid. Let us recap the high-level architecture quickly. So we have collected the disparate sources in a warehouse, and that hand, it shows a layer of a data science. But on a click, you have prescriptive, predictive, and descriptive analytics in place. Now, use cases achieved. These are the use cases which we have practically proven in a Caliber lab. The first use case. Yes, you are reading it right. This is not a typo. Annual product quality report has now become anytime product quality report. The reason, because data is integrated, you just give a date range and select a product and take out the report the way you want, be it a yearly, half-yearly, quarterly, monthly, or maybe daily. Second, of course, the time and the manpower, which is getting engaged for months together to have this APQR in a place, that is solved in a minute. Data storage, as I have told you, that because of its commodity, historical data is not a problem. Hence, churning out the analysis out of the historical data is not a problem at all. The important thing in this case of APQR is it's a real-time product quality report nowadays because data is flowing from the island directly to the warehouse. And anytime, it's a real-time. Second, process capability and Six Sigma analysis. I understand that there are many statistical software tools available in the market which gives this. But believe me, without using that, we have developed this in 3 days. And this will give you out-of-trend summary, what is your normal distribution by Anderson-Darling test, the CPK and PPK. These two are the, of course, big-ticket items. Now for us, as this is only a lab data, this is a very small ticket for us because it is just extracting the data from the one system. And here, the beautiful summarization and the visualization for the sample analysis. The same test results, any test results you give and select the batches, or you don't select the batches also, that is also fine. Just select a date range, and it will give you the test trend summary results. Another, the proportion, how many sample analysis we have performed, how many completed, and how many it is pending. And the above one is a matrix report. The column, it is of a date range, and the rows, they are of a sample type. Once we click on the intersection of a cell, you may drill down again to find out the details. Another, it is from a QA, another matrix. What are the issues? What are the kappas? What are your permanent change control across the plant? And how many numbers? Again, substantiated with a beautiful visualization. Quality metrics is another big ticket item along with anytime product quality report and a process capability. This quality metrics, we have not completed yet. We are in a process of completing it. Very soon, probably in my next session, I will be talking more about the quality metrics. Now, next phase, machine learning and AI. Believe me, these two words, they are not a big buzzword nowadays. They are the trend. And that we have seen with what Dr. Jimmy, it was just a mind-boggling session from Mr. Jimmy. And let us see what are the use case for the machine learning. Suppose a sample is there and my machine, or for example, it is algorithm predicts the outcome of sample analysis with a confidence level of 85 to 90%. How about that? Or machine failure detection by algorithm. How about that? Exciting, right? Now, the artificial intelligence is a complete ball game altogether. In the event of a machine failure, if that algorithm suggests us what is to be done, I guess it's a speechless, right? So that is the power of artificial intelligence. And these are the use cases we will be doing very soon once we finish our quality metrics. Now, I know that this is a very, very long journey. So I would like to conclude my session by giving you 3 highlights and 3 takeaways from my session. First, develop a strategy and vision to accomplish a clear analytical objectives. Second, take a small step, baby step. learn, evolve to complete the entire pyramid. Last, not the least, to bring the organization culture for data analytics and data mining. The reason for having that culture in our organization is because a big potential wave that is a deep learning is going to come very soon, and this time, along with other industry, pharma industry should also be welcoming it. Thank you so much.
Thank you very much, Dr. Kavita. It was really interesting talk. And that's also the end of first session. And I think the message is very clear from all the three speakers. Okay, the paperless lab, not a dream, not a reality, but a necessity, no choice. Okay, AI and machine learning definitely is required to make the process faster. And finally, data is a new oil. You refine it with data analysis and you get better results. Okay? So I'm sure this is very interesting session for all. And we have the audience from different countries with different regulations, very advanced regulations like US and Europe, China and India who are catching up the regulation due to high export. At the same time, we have from Southeast Asia and South Asia, where the pharmaceutical is still growing. And I'm sure this session must be very useful for all of you. And you may have many questions, but due to time, maybe we can ask a quick question if you have for any of the three speakers. Please ask the question. I think we can have only one question. So, first one first. Okay. Anyone? Or everybody is very thrilled with it? Okay. Actually, I have received one question for some of the customer who don't want to declare. Okay. And this question is for Mr. Indrajit Bose, but I think Dr. Kavita also can answer. So there are so many softwares in the lab and networks, many networks, which need data output, okay, data input output, sorry. So how do we ensure compliance and quality with so many different file formats? So who would like to answer? Yeah, Mr. Bose.
Hi. Hello. Yeah. It's a very interesting question. On reality, actually, there's so many softwares available. And that's, if you see in my presentation, I have told that one of the very important aspects is that you'll have to have three things. One is that a very, very clear roadmap that what you are having it today and what you are going to have it, number one. There is no standard answer that this is acceptable, this is not acceptable. Second thing is that you have to have the skill and the expertise to understand what is the requirement of the regulation. For example, you have to have very, very good knowledge on CSV. Many times in the organization, we do not have that. If you do not have that, in fact, today, there are so many experts available to guide you on the CSV. There are many, many contracted agencies also, if you do not have internally, because you cannot develop every expertise within the organization yourself, because that's not your core job. Your core job is to manufacture testing of a product. Your core job is not to develop an IT solutions. So three ways I like to tell you that first, you have a vision. Second, if you don't have the capability to assess that this is the best solution for you, go to the experts available in the market. Understand what is the CSV regulation. Then with that understanding, assess the vendor and select the vendor based on your requirement. And absolutely there is nothing standard that he is good or there is bad. It is not like that. Thank you.
Thank you very much. And I think with this, we can wrap up this session. If you have questions for any of the speaker, feel free to contact them during the networking break, during the lunch. You will have enough time. And now, from the data science, let's move to the food science and take a refreshing break and come back at 10:55, please. Thank you very much.
Yes, I hope so.
Yeah, hello, ladies and gentlemen. Thank you for being back to the second session. I'm the moderator of this session. My name is Jotolf Axleben. I'm coming from Shimadzu, Europe, and I can say I'm glad to see so many customers and so many interesting people here in this room. So I really enjoy. Thanks, friend, for the invitation. So the next session, technology of tools. After we talk a lot about data, now we like to talk about how the data are taken and what technologies we have nowadays to support your work in a proper way. And we will hear a lot of interesting stuff concerning separation science, of course. And we have a couple of nice and very experienced speakers for the session. So it's my pleasure to announce the first talk given by Dr. Baoning Su, Vice President, Core Analytical Services, Fuji Application, China. So thank you for joining, and the stage is yours, Dr. Su.
Can you hear me? All right, so from AI to FI, now let's switch gear to TI, okay, technology intelligence. Thanks very much for Shimadzu for the invitation to attend this wonderful event. This is also my first time to Singapore, so wonderful experience. Today, I'm going to give three discussions. So first, very brief introduction for analytical team at Wuxi AppTec. Actually, this is only part of analytical. And then, SFC separation, what capabilities and what work we do. And finally, will present, actually, the work use SFC for drug discovery and development. What's the key considerations? I'll give you a few examples. So here, as many of you know, WuXi is an open access, pharmaceutical research, technology platform. So this is research service division. So at the top, so research service division, we have total 9 business units. So at the top 5 is chemistry related. So the first CTD is mainly for the training. IDSU is the International Drug Service Unit, mainly collaboration model is FTE. We have a lot of collaborations with Europe, US, and many other countries. CSU is project-based, okay, called Chemistry Service Unit. So DWL actually is DNA encoded library. It's kind of very attractive new drug discovery technology. At the most right is CAS, Core Analytical Service. So this is the team I'm leading. So analytical team to mainly support all the chemistry teams, the entire discovery chemistry of the WuXi. So total we have 6,000 discovery chemists at WuXi AppTec. The bottom 4 are the biology-related business unit. So core analytical service, as I just mentioned, IDSU, CSU, and actually we have one more, chemistry business unit is called DDSU, Domestic Drug Service Unit. Mainly collaboration is for domestic Chinese companies. So those 3 business units total 600, synthetic chemists. We do complete, analytical purification, structure identification services to support those chemistry teams. Also, WuXi, we have process and manufacture called STA. So STA, they have their own very huge, like 600 people analytical team, but my team also do some support for STA as well. In addition to internal support, we also do a lot of external projects, collaborate with domestic and international, clients. Use the areas of analytical purification, structure identification. So right now, total, we have 480 analytical scientists under my team, and lab space is 12,000 square meters. The team located at 5 sites, including based in Shanghai, Qidong, Tianjin, Wuhan, and the newest site is in Chengdu. So here, for analytical, as many of you know, instruments are very, very important. So these are just some of the major instruments we have. So like NMR total, right now we have 32. This is not up to date. Actually, now it's 36. So in different sites, this is the total number. So all the analytical purification and some special testing instruments. So total, we have 500 state-of-the-art instruments. So this slide just show you what kind of services meet what work we do. So first is high throughput purification. We have purification, milligram prep HPLC and large-scale prep HPLC, total 160 instruments. So annually, every year, we do like 500 to 700,000 compounds purification. Structure confirmation identification mainly use mass spec, ADYD, and 2D NMR data. So physical chemical property determination, log D, log P, pKa solubility, we have a dedicated team to support our chemistry teams. So today, I will focus on only SFC color separation. Mostly we use SFC, but occasionally we also use HPLC to do color separation. So impurity isolation identification to support, mainly is for the late stage scale-up project and process, development. Also, we have lab to provide GMP-like regulated service. So including method development, validation, sample release, stability testing, et cetera, et cetera. So analytical team also, we always are trying to establish our new, skills technologies like ASMS, affinity selection, mass spectrometry. So this is, one of the new techniques we just established very recently. So also, we have a group located in Tianjin to provide natural products-related services, including extraction, isolation, and identification. So in the right side, just some common analytical purification samples every year, how much we handle. So like, we have so many instruments I showed you in the last slides. Like LCMS this year, so we do like 4.5 million samples. So many, because of Wuxi, we believe, we are the largest chemistry platform, global-wisely. So analytical and purification also, we have very, strong capabilities. So the next section, I will just, discuss some, the application of SFC for drug discovery and development. So this is, many of you, including Chris, is the expert in this area. So SFC, of course, has a number of advantages, regarding to actually not only color purification, but color purification versus, widely used reverse phase HPLC. So it's great technology. We know, majority of the mobile phase is CO2, only use limited alcohol solvents. So fast mass transfer during separation. So that's why, the separation efficiency can be much better than HPLC. A color separation for HPLC, many of you know, the method might be 20 minutes, 60 minutes even. SFC mostly is 3-5 minutes. So free of water, no water, many compounds are sensitive, not so stable with water. And plus, if large scale, if you purify the compound, use a reverse phase, you get a huge volume of the aqueous solution. The workup is also huge challenge. So short raw time, I already measured mostly very short. So that's give you high speed of the purification, and obviously cost is more efficient. So next list, what kind of SFC instruments we have, analytical prep. So SFC not like maybe HPLC at LCMS, limited vendors to mix the instruments. So basically, all vendors, we have the instruments. Total, we have right now, analytical, semi-prep, and prep, we have 45 instruments. So this means over 50% of the SFC instruments in China are located in Wuxi. So we do have analytical, Shimadzu, Nextware UC2 analytical system. It's great. So far, didn't use prep yet, but we have closely, working with Shimadzu China sales representative like Dawei and Yingchong give us very strong support in the past years. Very soon, I think Shimadzu, prep SFC, will be our lab. So this slide shows, typical workflow at Wuxi Tech. We use our SFC platform. So when we receive a sample, first we'll screen, use SFC to screen, which column gives the better separation. So here, we didn't use AI yet, but this is also AI today learned from this summit is analytical intelligence. So we put an instrument first, just screen those 8 columns. Why those 8 columns? Because in past years, every year, we do like 40, 50,000 analytical samples at 10,000 plus separation samples. Based on the experience, those columns give higher success rate for the separation. So that's why now first the screening, we just use those columns. When we get the condition, then we'll move forward to the prep isolation. If no separation, then we are screening use other SFC columns. If still no separation, then we'll use HPLC. Occasionally, some challenge samples, you cannot get better resolution, use either SFC or HPLC, then we may need to consider to do chemistry modification. So this slide shows typical what we like. So this instrument actually it can really help us to improve method development efficiency. So one sample in each injection, 8 columns run parallel. So in 10 minutes, you will get the separation for one sample in 8 columns. For example, the first sample, use methanol 1 injection, ethanol 1 injection. Clearly, immediately, in 20 minutes, you will know the first column gives much better separation, and the ethanol mobile phase is better. So this can really, we like this instrument a lot, but the bottleneck now is this instrument has higher instrument downtime failure time. So the second sample, for example, the fourth column gives better separation. Of course, as I mentioned, not all the samples SFC can give you good separation. For example, the third sample, we screened those all 8 columns, no separation. And other SFC columns with after screening got separation, but on the analytical only baseline separation, the resolution is not so great. So we further screened use HPLC. This separation is much better. So this is the typical workflow we do at WuXi AppTec. So what the services we provide use our SFC platform. So first, of course, method development. When we receive a sample, we will use either SFC or occasionally HPLC to develop a method. And there's separation from milligram scale to multi-kilogram scale. So far, the largest batch we did color separation is 130 kilogram for a single compound. And then, of course, determine the color purity, EE value of the isolated compound. And sometimes, after color separation, we need to study its stability. So racemerization under different conditions, see whether the compound is stable. Mostly, we use SFC to do color separation, but also some color separation samples we use SFC, like the advantages I already measured use SFC. So again, right now, so we do analytical sample every year. Now is like 50 to 60,000 samples each year, and actual separation sample is 12 to 15,000. Of course, that number is just roughly, because if we have a few really large-scale samples, so that number maybe will decrease. Then give you just some basically. I don't want to routine samples. Don't want to show too many examples. So based on our experience, you passed close to 20 years for SFC separation, because we support not only discovery, but also development. So what's the difference consideration for drug discovery and development? For discovery samples, because most important is the speed. It's for only limited quantity needed, only a few million grams. So the speed we are talking about is hours, okay? If we can deliver the sample to the chemist, a few hours, faster, that's, they care about. But for the development, generally is large scale. So mostly is for toxicology study. So this is really when they have no choice, because we know chromatography purification is the most expensive, technique, right? So only when they have no other choice. So the speed, not hours here, maybe just days. The speed is also important, okay, for toxicology study, yeah. But not as critical as for the discovery samples. But for the development, another concern is cost, because generally large scale. So we need to, for example, so this is one discovery sample. If the method, we can isolate the desired compound with enough purity, that's okay, just baseline separation, because only a very limited sample. But for the process, if we multi-kilograms, that method absolutely, we will spend a few more hours or even days to find a better method, because the overall cost will be huge different. So development, like we need to get completely, well-resolved, method. So other considerations, very important considerations for SFC separation. I'll give you here two examples. One is this compound B, for example. This compound contains amine group. So under SFC conditions, we screened many different conditions. Still, you will not baseline separation per peak shape. So this is no way to proceed for prep isolation. So then, what we did is you can again do chemical modification. So this compound simply just do the bulk protection. After bulk protection, see the separation peak shape is totally different. So makes the separation so much easier. Another consideration during actual application to purification. I mean, this is maybe not only for SFC, even for HPLC, similar logic applies. So for example, we received a project in step 7, this step. So after screening, got a reasonable separation condition, but not so good. Again, the resolution is not great. And then we asked chemists to get earlier intermediate, like step 6, 5, see the separation is totally different. So this is to determine, so also the throughput and the cost is very, very, because at Wuxi, every year we had so many samples, so many different projects. So this is also, many cases every year, we have this kind of situation. So that's for the SFC, mainly, as I said, we use for the color separation. Occasionally, we do the color separation as well. Color separation, especially for the water-sensitive compounds and for the large-scale purifications. And beyond that, also sometimes SFC for the radioisomer isolation, similar to color separation, can be also very, very challenging. So here are 2 examples. For example, this radioisomer mixture under HPLC condition, just very sharp white peak, but use SFC, you can easily get 2 isomers resolved. Another example similar, okay, also radioisomer. HPLC white sharp peak, very good peak. SFC, you can get separation. So I mean, SFC, this is really very, very beautiful technique, okay? So we use it a lot. And every year we try to establish, we get more and more samples. So even we are considering maybe use later discovery chemistry. Right now, the purification, typically we use reverse phase HPLC. We may use SFC to purify those compounds as well. So basically, so this is the information I want to share with you. Thank you very much for your listening.
Yeah, thank you very much, Dr. Su. I'm very sure you have a couple of questions, so please don't forget about them. We will have some question and answer session after the 3rd presentation in this round. So, talking a little bit more about drug discovery and supercritical fluid chromatography, I'm happy to announce the next speaker, Dr. Murali Taran, and he will talk about SFC, an accelerating edge in drug discovery and development. So I'm very encouraged to listen to your talk, Dr. Murali Taran. So please, the stage is yours.
So good morning all. As Doctor has already covered the basics of SFC, I would like to go more about on applications of SFC chromatography.
Gentlemen, this is normally the point where you get some music in television in case that something happens, so I hope we will figure out soon.
Yeah. So today's topic will be on SFC and accelerating aid in drug discovery and development. So to begin with, we always talk about safety. I'm sorry. So the outline of the discussion, so today's discussion will be how SFC is faster than chiral HPLC, and what are all the advantages of SFC over regular chiral HPLC, and the economics on the SFC purifications. So before we start, let us see some basics of the SFC. SFC works on the fluid CO2. So how this fluid CO2 is attained? Fluid CO2, at its critical point of CO2, the liquid CO2 will become the fluid. So fluidity is attained at the critical. So what is critical point? So we all know that critical point above which you will not have the substance in any of the three phases, neither gas, nor liquid, nor solid. So for CO2, the critical point is attained at 31 degrees centigrade and at 74 bar pressure. So at this temperature, the liquid CO2 is becoming the fluid CO2 and it is becoming the supercritical. But many of the publications, if we see, the CO2 is, SFC is being operated at its subcritical stage itself. That is below 31 degree and below the 74 bar pressure. So if we mix any of the two solvents like a water methanol or water ethanol, the back pressure, and if we inject into a reverse phase HPLC, the back pressure increases. Whereas if we inject a CO2 and with ethanol or methanol, the back pressure is not that much compared to the water, methanol, water, ethanol into the HPLC system. It's almost 3 times lesser than the reverse phase chromatography. So the same thing it's explained in the Van Demeter curve also. Sorry. Van Demeter also. So Van Demeter, so here, when the plot is plotted against the theoretical plates versus the linear velocity, if you see for HPLC, when you increase the linear velocity, the height of the theoretical plates increases. Whereas for SFC, even at a 0.6 flow also, the theoretical plate height is not increasing. So thereby, we can operate at efficient flow and with efficient pressure. See, based on this, whatever we have learned, we can go into the resolving power of SFC. If you see here, we have injected one of the polymer. So when the polymer is injected, the numbers what you are seeing 20, 38, and 100, they are all the MERS, number of the MERS. How well it is resolved in the SFC technique, we can see. So as Dr. Su has said earlier, the chiral analysis takes in hours. Later, it has come down to minutes, 30 minutes, 40 minutes. Then it has further come down to 3 to 5 minutes. Now the technology has improved. Now we are able to analyze using even in a 42 second, less than a minute also we are able to analyze using the SFC analytical techniques. See, in general, I have taken this small compounds, spiro compounds. In general, small compounds will not retain in the column. It will come out immediately. Whereas in SFC, when you inject it, because of the diffusing nature of the CO2, it penetrates into the cavity of the column, into the stationary phase, it retains, and then it comes out. That is from mobile phase to the stationary phase to mobile phase, it is coming out. So if you see here, so here it is by chiral HPLC technique, whereas here it is an SFC technique. How well it is resolved, you can understand, and the runtime as well as the resolution also better compared to the chiral HPLC. So this is another case study wherein the heterocyclic compounds. Heterocyclic compounds, you all know that it is mid-polar compounds. But when we introduce pegylation to that, it will become polar compounds. So the polar compounds, when you inject into the chiral HPLC, generally we have to dissolve in some polar solvents. When you introduce into it, so again you are not getting the good separation in the chiral HPLC, whereas in SFC we are getting the better separation. So that is one of the case study for the pegylated heterocyclic compounds. This is another example, macrocyclic compound. So macrocyclic compounds, generally, they are having the poor solubility in nature. So in chiral HPLC, because of the poor solubility, it will not come out that easily. It takes a longer time also. Whereas in SFC, when we introduce, when we inject this macrocyclic compound, we have a setup called pressure gradient system. Using the pressure gradient system, we can increase the flow as well as increase the temperature of the compound, temperature of the mobile phase. By that, we are getting the better separation in the for the macrocyclic compounds. So this is another case study, nucleotide phosphatic esters. So generally, nucleotides are highly polar compounds. So when we introduce an aliphatic chain, long chain phosphate ester compounds, the compound becomes the non-polar. So non-polar compounds, when you introduce into the, inject into the chiral HPLC, it elutes fast. And, but again, if you see non-polar compounds, we have to dissolve into the non-polar solvents. Whereas in SFC, the mobile phase liquid CO2, fluid CO2 itself is an act as a non-polar solvent like N-hexane and N-heptane. So by that, we can inject into the SFC and we'll be getting the better separation. So this is another interesting example. This compound boronic ester, it has come for the analysis to the analytical department. When this compound was injected into the LCMS, actually the idea for the chemist is how much of boronic acid has converted into the boronic ester. So that was an idea. When the chemist has given the sample, initially we injected into the reverse phase LCMS. We have seen that both boronic ester as well as boronic ester was present, acid was present in the compound. We were under the impression that the boronic acid has not converted, into the boronic acid. So the reaction was extended further. Then even then when we inject it, again we were seeing this boronic acid as well as boronic ester. So then again we have checked in the NMR instrument. So NMR also we were seeing the boronic acid as well as the boronic ester. Then when we all went and discussed with the synthetic chemists, there we found that boronic ester, when it contacts with the aqueous solvent, it converts into the boronic acid. So that the same sample, when we injected into the SFC technique, there is no aqueous in there. We could get a single peak. Thereby, we confirmed that the boronic ester has not converted into the boronic acid. Further, it was checked with the mass connected the SFC instrument. There also we have confirmed the mass, and we have confirmed that there is no boronic acid present in the compound. So this is another case study wherein when the sample was injected, actually this reaction was an enzymatic reaction. Enzymatic reaction means one isomer will be removed. So this compound, when it was given to the analytical department, we found that when we injected in SFC, we were seeing 4 peaks. We were under the impression that the enzymatic reaction has not happened. We were thinking that all the 4 isomers were present in it. And when we checked the UV also, the UV is almost same for all the 4 peaks. Then when we injected into the mass coupled SFC instrument, we found that the first peak is an impurity present in the compound. That was confirmed by the mass of that peak. The rest, all the 3 peaks were having the same isomer, and we have confirmed that enzymatic reaction has happened in that reaction. So this is another advantage of the SFC instrument over the chiral HPLC instrument. In chiral HPLC instrument, we cannot do the stacked injections. Whereas in SFC purification instrument, we can do the stacked injection. So here if you see, we have purified 2 kg of the compound within 10 days of the SFC time. Whereas if you are taken in the chiral HPLC, it might have taken a longer time because we don't have the stacked injection in the SFC instrument. So finally, the economics of the SFC over the chiral HPLC, it's a superior chiral chromatographic system, faster retention time and smaller run times, high throughput screening and purification timelines over HPLC is better, optimum on-column loading capacity enabling the faster purification, and as you all know, it is a greener chemistry, and majority of the CO2 can be recycled and then we can re-inject into the system. And this is a comparative study between the HPLC and the SFC. We have purified some 780 gram compound in 15 hours of SFC time. Whereas, of course, this data is extrapolated. Whereas, if we would have taken in the HPLC, chiral HPLC, it would have taken around 85 hours of time. So that's the way the SFC is a better technique compared to the chiral HPLC instrument. Thank you.
Yeah, thank you very much, Dr. Murali-Tharan. At least a lot of reasons why to consider about SFC nowadays. So when I see a lot of these chromatograms, I feel I did something wrong doing HPLC for many years. But you never know, things are changing. So now we're coming to the final talk of this session. I'm happy to announce Dr. Christopher Welch, Executive Director of Indiana Consortium for Analytical Science and Engineering and founder of Welch Innovation LLC US. And I can also say some part of our collaborators in terms of ETC, Enabling Technology Consortium. And in this regard, I'm happy to announce Chris Welch here. Please.
Thank you. Thank you. Okay. Well, it's great to be here. Thanks so much. I think that these events where we bring people from across pharma together to sort of study where we're at and plan for the future are really important. So I'm very honored to be a part of this one here today. I'm going to talk a little bit about online analysis and inline testing. This is a topic of growing concern in the industry. We'll throw in a few additional topics as well. So here's my biography in one page. So I started in 1981 as an undergraduate working on chiral chromatography, and I've done lots and lots of chiral chromatography and preparative chromatography over the years. I worked for Merck for many years. I've been working in high throughput analysis and high throughput experimentation. This is a fun area for me. For 15 years at Merck, I ran the New Technologies Group that's seeking out and bringing in, first into the company, research technologies. Not new drugs, but new instrumentation and tools of that sort. We had a budget of over $25 million a year at the peak capacity, and I've done over 300 projects over the 15 years, a lot of that bringing in new instrumentation. That's something I really enjoy quite a bit. Since retiring from Merck in 2017, I've been keeping busy doing a lot of interesting projects. One of the most interesting and gratifying is working on the development of the Shimadzu Nexera PrEP SFC. It's the new PrEP SFC system that's just recently been introduced and that won the 2019 Gold Excellence Award at PitCon. I'll circle back and I'll tell you more about this later in the talk. But other things I've been doing since retiring from Merck, my wife and I started Welch Innovation, and I've started working with a number of small companies, and I'm advising them and have part ownership in several companies, including Snapdragon, a flow chemistry company, and Enantiosense, a sensor company for enantiomers, and Novolytic that is looking at some online analysis technologies. I've also been working with Notre Dame, Purdue, and Indiana University to sort of play a role in the innovation in the university environment. Okay, so kinetic profiling of organic reactions. It's increasingly important, and I think a lot of us have done this in graduate school, perhaps where we're doing a reaction in an NMR tube and we're watching starting material convert to product over time. We can study this with a lot of spectroscopy techniques, is what has been done historically. Here's a react-IR approach, or we can look at UV. But increasingly, HPLC is being used here. So simple experiments here can reveal the reaction order and shed light on reaction mechanism. So this is very important. If we really want to understand the reactions that we're doing, kinetic profiling can really provide a way to do that. And it's increasingly used for monitoring continuous processes. Ideally, it's best to use simple-to-use analytical instrumentation, because a lot of times we have to have our synthetic chemists and engineers performing the experiments. And fast analysis cycle time is important if we want to study fast reaction processes. And as I said, HPLC and HPLC-MS are gaining. So here's some old school kinetic profiling experiments. I've done a lot of these over the years with spectroscopy or CD, or a combination of HPLC and CD. And you can look at, for example, the enantiomerization of this compound over time and see it going to racemate as we do that. Or similarly here, we have restriction rotation around this bond here. And there are two enantiomers, and we can isolate by PrEP SFC, as we just saw in the previous talk, put it in a bottle and watch the decay of enantiomeric purity down to racemic over time. We can combine HPLC with complex analysis types, for example, ICP mass spec, to study some pretty unusual things. In this case, we're looking at the formation of this dirhodium catalyst, and we're using rhodium ICP-MS combined with HPLC to look at all these intermediate species in the preparation from rhodium tetraacetate all the way to the desired rhodium tetramepi product. Just to point out, it's sort of interesting, there was a big puzzle with this reaction. You needed to cook it in order to get the product, but no one knew why. But if you see here, there is a mismatched tetramer here that is the undesired isomer, where everything is correct except we have one thing backwards. And we need to cook it and for that kinetic product to turn around and thermally equilibrate to form the final desired product. So additional eyes into the reaction brings understanding, allows us to bring control to the reactions. I worked on the development of this instrument some years ago, which I think was a little bit ahead of its time, but I think we need something like this coming forward. This was a beautiful instrument, microflow, HPLC. You could wheel it up to a bench. It withdraws aliquots over time. You can quench, you can dilute up to a factor of 100, do chiral or achiral HPLC. You can vary the sampling interval from 4 minutes up to hours, and it runs at microliter flow rate. So here you can see starting material is gone. We see an intermediate growing in and then disappearing, and finally product formation. And you can take this kind of a tool and put it on a flow chemistry reaction. And with flow chemistry, it's all about the rate at which the reagents are flowing through the reactor, so the residence time, and also we can imagine the temperature inside of this reaction space is also important. So very easily you can set things up to vary residence time of the flow rate and to vary the temperature of this chamber, and you can go through and do something about understanding what the isomer ratio here is as a function of temperature and residence time. And you can see exactly what's needed to drive the reaction to the stage where you need it. There's a lot going on in this field today. It's maybe too much to summarize, but some of the interesting players here, Jason Hine at University of British Columbia and his group are doing lots of interesting things with online analysis. So if you're not familiar with what Jason's doing, take a look at some of the papers coming out of the group. Here's the Abbott group with Steve Doherty here, and Darshan Patel is actually the guy who's been working on the instrumentation here. But lots of pharma companies are digging in and doing their own home versions of online kinetic profiling. And here's Graham Cook's doing simple mass spec, no HPLC here. We're just hooking a mass spec up, and we're pulling sample from the flask in and doing a direct mass spec. You can see that there's quite a bit of noise here, but it works somewhat. Obviously, it's a little better if we do it in conjunction with HPLC. So lots of interest coming up in the field, and I thought it would be worthwhile to cover that. Interestingly, the chromatography is speeding up. We saw our previous speakers mention this. You know, when I started here, typical separation might be 10 minutes to resolve something. That kind of separation that we could do in 1980, we can now do in 3 seconds, right? So because of small particle size and the evolution of column technology and et cetera, everything is much faster. This article in ACS Central Science tells the whole story of that over time, but the bottom line, for example, for chiral analysis is almost every racemate can be separated in under a minute, and many of them in only 5 seconds, something like that. And so it's a different world than we lived in just a few years ago, and that's very useful for online reaction monitoring. I'm going to, let's see, I think I will. Well, yeah, and it's a good thing because if we have a chromatography analysis time of 5 seconds and our autosampler takes 30 seconds between cycles, then who cares, right? So it's interesting and useful that the Nexera autosampler is so fast, right? It's the world's fastest single needle autosampler. And that's very important because it allows you to keep up with the speed of chromatography and exploit the advantage of being able to do a very fast chromatographic separation. Let me tell you briefly about a technique called Miser chromatography. This stands for multiple injections in a single experimental run. Miser is just like a flow injection analysis, except everything is being chromatographically resolved, right? We're using a column. It's a little short 1 centimeter column, and we're injecting a sample from well 1 and then well 2 and well 3, all within one run, right? And we're just barely separating the compounds of interest from the interfering compounds like solvents and salts and bases and things like that. And then we're using mass spec to look at the signal for the starting material and the product, and we're doing a reaction screen analysis. Now, normally when we do a microplate evaluation, we are doing the chromatography, we're integrating, we're taking the integrated area counts and putting in an Excel table, and then we're graphing those and we're making a decision about what's happening. With Miser chromatography, we're just looking at the graph. We're not integrating, we're not putting anything in Excel, we're not graphing. The chromatogram is the graph, right? So if we start with just starting material here and no reagent, of course there's nothing. Now we start adding the reagents or the catalysts to see which are working, and we can see here conversion to product. We have a little here, but this one's very interesting. Lots of product and all the starting material is gone. Here's another good one. So we can just see with our eye who the winners are. So speed to decision is much faster, right? It's not fancy, it's very simple, but it's a nice usable tool. Miser chromatography is also very useful for kinetic profiling. Let me show you a couple of examples. Well, first, let's just show that the Miser works very well with Shimadzu software. And in this case, we're doing a SFC chiral analysis here, and we're injecting every 10 seconds perhaps. And you can see we're doing 96 wells in only 33 minutes here. And the nice thing here is that we can do it with only 17 lines of code to command the autosampler. Because what we're doing here is sort of a repeat cycle, and we're telling the autosampler to go to the next one, and then wait, inject, and then wait a few seconds, and then go to the next one. And so you can make like a loop function, and you can, in 17 lines of code, describe everything that you need to do. If you try to do this with Agilent autosampler, you need like 5,000 lines of code for a 96-well plate, because it doesn't really have the loop function characteristics that are in the Shimadzu software. Kinetic profiling using this approach. Here's a couple of examples. This is from Hammett, 1937, the physical organic chemistry, the Hammett coefficients and so on, where we are seeing an electron withdrawing group here speeds up the reaction and electron donating slows it down. wardrobe malfunction. So we can see here, here's the starting material decaying and now the product coming up. So you don't have to graph anything. Again, we can see automatically the kinetic profile of the product formation. This keto group, we can see it's intermediate in reaction time, and this electron donating is very slow. It's just starting material and no reaction occurring. We can do this for some very fancy reactions. I think in the order of time, I'll push on though to show, here's a single starting material. We could get 2 different intermediates and 3 different products. And all of those have different masses, and so we can just dial in the masses here and see there's the rate of disappearance of starting material. Here's the formation of intermediate 1 and the subsequent decomposition. Intermediate 2 is much slower, and so less formed, and the formation of the 3 products over time. The most probable reactant, the mixture, and the least probable times 2. Okay. Here's an example of using online analysis using an easy sampler. Now this is a tool that's frequently used in pharma. It pulls samples out, puts them into these tubes, and then we analyze them later in an HPLC instrument. Well, you can also set this kind of thing up to do a piping right from this tool here. So instead of putting it into vials, we can just do the immediate online HPLC and get an online reaction profile. Here's a nice example of this on a mystery reaction from Merck. We found when we wanted to do a hydrolysis of this T-butyl ester that it didn't go unless we had methanol in the mixture. And so we hypothesized, well, maybe it's doing transesterification. Maybe that's the reason why methanol is needed to be there. So we had a hypothesis that maybe we form a methyl ester intermediate, and then that gets hydrolyzed to acid. So here you can see, we look at here, the starting material is degrading. We see the methyl ester indeed is growing in as an intermediate and then going out over time, and the product formation there. So we had a hypothesis, we do the online kinetic profiling, and it confirms that hypothesis. Miser is fun, you can try it. It's a good thing to teach. Here's some high school and college students playing where we're looking at the chili pepper analysis, looking at capsaicin from different kinds of chili peppers and hot sauces, and it's a fun thing to play around in your laboratory. I wanted to say a few words about this project that we did in a cross-pharma way to develop the ETC Prep SFC product, the Nexera Prep SFC. Now, I've worked a lot in bringing new technologies into pharma, and things have been changing over the years. And a lot of pharma companies used to want to do it all by themselves, but the world sort of changed, and the pharma companies are more eager now to work collectively to try to accomplish their goals. So here's a paper that we wrote about why we should work together. And my colleague from Pfizer and BMS and I from Merck put this together originally and said we should share costs and collaborate together. And out of this came a formation of something called the Enabling Technologies Consortium. I guess it's not on this slide here, but that does exactly that. It serves as a body to coordinate pharma companies working together to do development projects with external providers. So one of the first things that we did when we formed the Enabling Technologies Consortium is go after a very long-standing problem. And that was replacing the aging Berger multigram Prep SFCs. Let me explain what this was. This is something everyone in pharma was complaining about for a number of years. So Berger had a very beautiful Prep SFC instrument. It's what everybody loved. All the companies bought these instruments. Everything was great. Berger was very successful. Now, as a consequence of that success, they got bought by a company called Mettler-Toledo, and Mettler-Toledo then sold them to Thar, and Thar was then sold to Waters. And along the way, the company changed the way they were making this beautiful instrument. And the thing that everybody loved was no longer there. And so the companies were faced with this challenge of, this thing doesn't work anymore, and in fact, in order to keep it running, I've got to go to eBay to buy spare parts. I think this is just not working. Okay. Can you hear me? Okay, good. So we're going to eBay to keep these things running. And there are several things in the marketplace, but none of them are good enough. And so how should we, all the pharma companies, work together to get a better replacement here? Well, what happened was, we worked through the Enabling Technologies company, organization we got together, we defined the need, we requested a request for proposals, and a number of companies responded. They were all interviewed, and we selected a partner for the new technology development project, and that was Shimadzu. So the project was the ETC PrEP SFC project. Here are the companies that got together on that: Pfizer, Merck, Genentech, Amgen, BMS, AbbVie, Lilly, Biogen, and Boehringer Ingelheim, and GSK has recently joined in as well. This group got together and they said, in order to satisfy our needs, it has to have this qualifications, exactly what was needed. We met with Shimadzu engineers and design and software people to work through and they came with a prototype instrument, which is a little shielded here. And the bottom line is, it works beautifully. Here's the initial prototype, which we were very awed by, but the finalized product design is really a marvel. It's really a piece of beautiful work. Elegant, sophisticated design, very small footprint. It has no external chiller, which is a big noisy thing that's typically required for a PrEP SFC. There's none of those. It's just good design, got rid of that. Has a branch channel design gas liquid separator that's a marvel and really allows very fast isolation. Beautiful software. I think the major thing about it is the needs of the customer said it has to be able to do this, this, this. When you look at all that, it's mostly software. It has to be able to work in a streamlined fashion to solve the people's problems. And so it's a really very nice job of a method editor, instrument monitor, data viewer, modules of software that all work together seamlessly to get the job done. Performance is spot on. We have a paper that will be coming out recently that'll go through all of these data, but from a hardware perspective, everything works fine. These people put it through a big test, and everything is working well. Here's some typical separations coming from it: single injections, automated fraction collection, stacked injections. Here's isolation of a small impurity, and here is checking a minor impurity. So that's it. I wanted to quickly show you some of the ETC PrEP SFC stuff in addition to talking about online kinetic profiling, but happy to talk more about it with you at the break. Thanks very much.
Yeah, thank you very much, Chris, for your presentation. And I think at this point, we also need to address a big thanks to our research and development group, who finally did the work on the hardware on our side. And I would say this is also a reason to give a big applause to the colleagues, even they are not here. Yeah, now we have very limited time, but we have at least the possibility for one question. Who would like to be the one asking to the three guys about a question about the last presentations?
Okay, so Chris, may I ask, in your opinion, what will be the price acceptable range for a PAT, fully automated?
Yeah, I think it's a good question. I think, less is more is the smart aleck answer, but, less is always better. But I think, people would be willing to pay, I don't know, for the convenience of that, maybe as much as $200,000 for just a pure HPLC that really hit it well, if it had the convenience, right? Right? You're gonna buy 16, I think. But that being said, there's a lot of marketplace. Are they going to buy one? Or are they going to buy 20? right? And I think that you can imagine every synthetic chemist having a tool like this. And if it's $200,000 for every single chemist, they're not going to do that. So I think that the low-end play here, sort of a simple device that will get the job done and be easy to use for the general synthetic chemist, is maybe where the real value is at, because the numbers go up, if that helps.
Okay. Many thanks again for all the presenters. Thanks for your patience. And now we reach the point at the end of the morning session. So thank you very much.
Yes, ladies and gentlemen, very glad to announce that this year Shimadzu has received a Product Innovation Award for AI-enabled Nexera series. As you may know, Shimadzu is winning Frost & Sullivan Awards for last 5 years for various product innovations as well as highest growth in Asia. And today, to present this award personally, Dr. Satish Lele, Vice President Visionary Innovation and Head Consulting Frost Sullivan, is here. So please welcome Dr. Satish Lele on stage. And to receive the award, may I request Mr. Maruyama-san, the head Shimadzu Corporation's Analytical and Measuring Instruments Division, and Ms. Naomi Okazaki, General Manager of Innovation Center in Singapore, to conceptualize the branding of this blockbuster product. At the same time, Mr. David Chan, who is the Product Manager of LC in Asia, to please come on the stage to receive this award. Okay, thank you very much, Maruyama-san, Naomi-san, and David. Dr. Lele, can you please stay on the stage and say a few words about why Shimadzu is winning this award. Just hold on for a mic.
Hello. Yeah. So thank you very much. Just to introduce Frost & Sullivan to all of you, we are a global research and consulting company established since 1961. And we have been tracking different markets across the world. In fact, we track almost 35 different industries. And two of the important ones which are important for today's session are the pharmaceutical industry in the healthcare sector. And we also have our analytics program under the test and measurement analysis that we do. So we run what we call a best practices award program. And in this program, as we do our research, the analysts continue to seek companies who are excelling across different areas of business. It can be in technology, it can be in innovative products, it can be in market leadership. So a variety of different areas where companies excel and thrive in business. We run this Best Practices Award globally, and we did an event in Singapore as well where Shimadzu received this particular award at that banquet. So essentially the idea is to recognize best practices in the industry and to bring them together on a common platform. Now the only difference between our research is that we do not invite any nominations for our reward. All the research is done by our analysts. We identify the top 3 companies in any particular domain. We do a one-to-one discussion with each of these companies, and then we decide who is the winner in that particular segment. So it's not an award that you can win. You cannot propose that award to us. You cannot represent your company to us. It is only our analyst who would choose and finally decide who the recipient is. So that's the background of the Best Practices Award. And we are very proud to actually announce that Shimadzu was the recipient of the Global Best Practices Award for the Nexera series. And we saw some of the presentations today which really brought out the capabilities of this particular product. And this is the, the comment from the analyst, where Shimadzu's constant focus on excellence is evident with the global launch of its latest Nexera series. Its strides towards improvisation has helped it establish benchmarks in the laboratory instrumentations industry. Right? So a big round of congratulations to the entire team at Shimadzu and for their hard work and the continuous innovation that we have seen. We have also seen that Shimadzu has been at the forefront of innovative products over the last 3 to 4 years, where we have been running this program, and not only for this product, but also across different analytical products in Asia Pacific as well as globally. So with that, thank you very much, and I look forward to hosting the session in the afternoon as well.
Thank you very much, Satish Lele. Thank you very much, ladies and gentlemen, for your cooperation, your participation. Definitely, we expect more in the afternoon session, and we also hope to win more awards in future. Okay, with that note, let's take a lunch break. We are still on time, so you can enjoy the lunch for almost 1 hour. Please come back by 1:05 for the next sessions. Thank you very much and enjoy your lunch.